Yeah, hi, I'm Alex. I'm happy to give this talk today on high throughput approaches to viral drug discovery. So here are my conflicts of interest. And moving forward, the overview for my talk about two different topics. One's examining SARS-CoV-2 adaptation to drug. Another is our approach to multiplex drug screening. And so for the first project, I'd like to highlight that it was really carried forward by three amazing graduate students, Shoa Katani, Debbie Hung, and Bruce Culbertson, in collaboration with David Ho's lab at Columbia University. So a little bit of background on COVID-19. I'm sure this audience doesn't need it, but just always like to make sure we're on the same page, thinking about the problem the same way. According to the CDC, COVID-19 caused 6.3 million hospitalizations, 1.1 million deaths in the United States. Vaccines obviously transformed this disease course, predicted to have saved 3.2 million lives, but yet not everyone's vaccinated. 5% of Americans over the age of 65 still have not received a single dose of vaccine, and not all patients respond to vaccination, in particular those that are immunosuppressed. And the virus continues to evolve to escape immunity. And so this has decreased vaccine effectiveness, and it's led to us needing to find alternative ways to fight this virus. So other options exist. Well, there are several chemical inhibitors available that target SARS-CoV-2 viral proteins. In particular, nirmatrelvir, which is marketed when combined with ritonavir as Paxlovid, is arguably the most popular. And that's because it's orally bioavailable and it's highly effective in their initial clinical trial showing 90% reduction in severe COVID-19. So how does it work? Well, it inhibits the SARS-CoV-2 3CL protease, and that's a protease that's required for the virus to make functional proteins. So quick refresher, the virus makes one very long polyprotein that then gets cleaved into multiple different pieces, and that's what the protease helps it do. And so what's the motivation for our study? Well, we knew that the viral spike had this incredible ability to mutate, and that had made vaccines less effective. We asked basically the question, does the 3CL protease have this incredible ability to mutate? And if it does, are there many routes by which it can gain resistance to drug or just a small number? And so how do we study SARS-CoV-2 adaptation to drug? We took SARS-CoV-2, we grew it in increasing concentrations of nirmatrelvir. And so you start with the virus here, you add it to cells, add nirmatrelvir, and then you let the cells select, so let the virus select for variants that are resistant to a low dose of drug. You then add the virus back to a higher dose of drug, again, let it select, higher dose of drug, let it select, higher dose of drug, let it select. And ultimately, as you finish this process, you've selected for variants that are quite resistant to the drug you've been treating them with. And so while most teams tend to do these viral passaging studies for about 2 or 3 isolates at a time, we wanted to really get this global view of how the virus might adapt. And so we had 53 isolates that we sequenced at 4 different time points to understand how the virus was adapting to this drug pressure. And so this is kind of a summary of all the different mutations that we saw, and I'll sort of walk everyone through those data. And so when you start with the wild type sequence and you start to look at the mutations that emerge first, you see these. And there are a few mutations that seem to happen quite commonly very early on as the virus is adapting, and that we see in these different lineages. And those are T21I, P252L, and T304I. Then if you continue to passage this virus in higher and higher drug concentrations, you see the emergence of secondary mutations and even tertiary mutations and quaternary mutations. And so when you look at that data as a whole across all the different isolates, you see that there are some mutations that arise early, such as P252L. Basically, this one kind of always happens early, or you don't really get it happening in another point. And some mutations always kind of happen at the end of the game. And so an example of that is usually like L50F. And so this suggests that there are some mutations that on their own are non-functional or perhaps even deleterious in the absence of other like enabling mutations. And so the other thing that we noticed when we looked at all the data is the virus seems to be kind of using the same mutations and just shuffling their combinations. So it's sort of like it has the same deck of cards that it kind of just plays out, but there's not an infinite number of choices it uses. And so that was this in vitro passaging experiment. So how do we know that the mutations that we found are actually the ones that are causing drug resistance? It's because while passaging the virus, it could be that other kind of weird, quirky things are happening that's allowing it to adapt to this drug. So to test that, we went back and we made recombinant viruses with each of the mutations that we had found from the drug passaging experiment, and we tested them against drug. And so, for example, you take the T21I mutation, you put it into the wild type viral background, and you see that it has about a 5-fold resistance to nirmatrelvir. You then do that for all these other single mutants. And again, we see in general, there's a small shift in drug resistance, although E166V is quite profound. And then as we start to look at like the double mutants we see and the triple mutants, we see this kind of additive effect where more mutations that we had seen in our studies leads to more resistance. And so that was kind of nice proof that what we saw from the passaging was likely real. And so we said, okay, well, that's nirmatrelvir, but do these mutations just cause some general resistance? Is it very easy for this virus to become pan-resistant to multiple compounds? And so we decided to test Antiviral, which is a drug that's in phase 3 clinical trials in the US, already pre-approved in Japan. That's a SARS-CoV-2 3CL protein inhibitor also. And so these are the data we got when we tested these same mutant variants against it. And what we see is that there certainly are some mutations that are in common, where if you get resistance to nirmatrelvir, you also get resistance to ensitrelvir. But we also found many cases where they're not overlapping. And so resistance to one does not lead to resistance to the other. And so again, as a whole, data suggests that we identified real resistance causing mutations, and that drug resistance doesn't need to be universal, that each drug can have its own little solutions to the problem. So next steps for this project, we've been monitoring SARS-CoV-2 clinical sequencing to see if some of the mutations that we identify are actually emerging in patients' data. But we so far haven't really seen anything strongly enriched. Last I checked a couple months back. But we have seen in the literature now the emergence of case reports of immunocompromised patients that have been treated with drug for weeks on end. And in those cases, they have emergence of resistance mutations that we actually do see in our studies. And so that kind of lets us know that there's some nice overlap between what we saw in vitro and what's actually happening in vivo in people. So summary for part one of our talk. We basically passage virus increasing concentrations of drug. We identify many ways by which the virus can get resistance to these compounds. And we found that mutations in nirmatrelvir can also cause cross-resistance, but it's not always universal to other clinical compounds. And so for that, I'll switch gears and talk about our second topic, which is multiplex drug screening. And this work was done by, again, 3 incredible graduate students, Debbie Hong, Shoa Katani, and Sam Resnick. And so a little bit of background on drug screening, which again, I'm sure this audience doesn't need. Typically, with drug screening, you start with a single target, whether it's a single viral protein or a whole virus, and you screen thousands of compounds against it. And this has obviously been incredibly successful, but it takes a good amount of time, money, and effort to get one answer. And in particular, the problem with viruses is that there are hundreds of viruses that currently infect humans, and there are tens of thousands of viruses that are circulating in animals, some of which have the potential to jump into humans. And so it's really, really great to find an inhibitor to something like SARS-CoV-2. Ideally, we'd be interested in an inhibitor that had a little bit more broad activity across a range of different viruses. And so what viral drug protein are we looking to drug? We're looking to drug the viral protease in our group, and we've chosen it because 50% of viruses encode their own protease. It's required for replication. There's been lots of success with a protease inhibitor to HIV and hepatitis C, and now also to SARS-CoV-2. And so our goal was really to identify broad-acting protease inhibitors. And to achieve that goal, we need to change the way that drug screens are performed. And so you need to develop some sort of method that instead of looking at one viral protease at a time, we'll look at dozens at a time. And so how do we do that? How do we screen for our viral protease inhibitors? We use a very simple chassis, which is yeast. And so it turns out if you take a little yeast cell and you express a viral protease inside of it, they really don't like it and they grow very, very poorly. You can then take these little yeasts and expose them to an inhibitor of interest. And if the inhibitor blocks the protease, then the cells grow much better and the cells are happy. And so this lets yeast serve as a very simple biosensor of protease activity. Protease active, yeast sad, protease inactivated by drug, yeast happy again. And so now it's very easy to make lots and lots of little protease models by just taking different yeast cells and giving them a different protease gene. And so that's great. Now we can make lots of models, but how do we test all the models at the same time? So to do that, we use DNA barcoding technology. And so here are an example of 4 cells, and here are 4 little DNA barcodes. And what we do is we take the cells and we insert into them these little DNA barcodes into a precise genomic locus that we know we're putting it into, and we know the sequence that every single cell is getting. And so going forward, when you see a different colored yeast in this presentation, it represents a cell that has a unique DNA barcode that I know the identity of. And so now what we can do is we can take the little blue yeast, and we can make them express SARS-CoV-2 3CL protease. The teal yeast can get MERS 3CL protease. The black yeast can get Zika protease. And the yellow yeast will get a control protein, EYFP, not a protease, yellow fluorescent protein, just turns the yeast yellow. They don't mind that. And so now if we induce the expression of all these different proteins, the cells expressing proteases will deplete from the population. They'll grow very slowly, while the cells that are expressing YFP, they'll grow really well. And so you see a skew in the population. And you can quantify that skew by exploding the cells open and just sequencing those little barcodes out and quantifying barcode abundance. We can now go back to that same initial population, induce expression of all the different proteins, but this time add a compound of interest. And if we got a result like this, where now the SARS-CoV-2 3CL and the MERS 3CL expressing yeast are growing much better, that would argue that this drug is a putative broad-acting coronavirus protease inhibitor that we would then investigate more. So does this barcoding idea actually work? So to test it, I'm showing you a focused study where we have a pool of 30 proteases that we tested against 96 compounds for a total of 2,880 tests within 196 well plates. And so I'll zoom in on a very small widget of the data. Here are some of our tested proteases within that pool. Here are some of the compounds that we tested the pool against. And a black square means a compound that rescued the growth of the yeast, and thus it's a putative protease inhibitor. And so basically, this experiment detected all the known interactions we were expecting. MAC5576 and GC376 are known 3CL protease inhibitors, where GC376 is known not to be very good against NL63, a 3CL protease. The lopinavir and darunavir are both HIV protease inhibitors, and they're rescuing the growth of the HIV-expressing yeast cell. And finally, glecaprevir is a known hepatitis C protease inhibitor, and again, it's rescuing the growth of the yeast-expressing hepatitis C protease. And as you also see, when you test different PARP inhibitors or kinase inhibitors, we're not seeing any effect. And so we're not getting a bunch of random spurious hits. And so what does our current assay look like? Our current pool actually has 40 different viral proteases encompassing 7 viral families. We also have a bunch of controls we've included in the pool that we use to sort of normalize our data out. And we've been screening that against a diverse library of compounds. And so here's a quick example of some of the data we've gotten recently from this pool. And I'll highlight these 3 compounds over here because they were basically what we were looking for. They're broad-acting coronavirus protease inhibitors, in this case against the 3CL protease. And so I'm showing you the structures of them on the right. And so that's great. These are hits that we got from this multiplex screen, but how do we know it's actually, real and it's not some artifact of the multiplexing? So to test that, we took the compound and we tested them one at a time against each of our protease expressing yeast models. And we did a dose response curve. And so this compound here has an EC50 of about 1 micromolar against the yeast expressing SARS-CoV-2 3CL protease. And the other 2 inhibitors, which again are very structurally similar to the first, also showed nice dose response profiles in the low micromolar range to the SARS-CoV-2 3CL protease. And when we tested this manually one at a time against all the other 3CL proteases, basically the results we got from one at a time testing mimicked what we expected based on the multiplex screen. And so that's great. We've rescued yeast growth, but how do we know, again, that that's specific to actually the protease or doing something weird to the yeast that's causing this rescue? So to test that, we actually purified out SARS-CoV-2 3CL protease, and we tested each of these compounds against the purified protein and did dose response curves. And what we see is that the compounds against purified proteins show kind of mid-nanomolar activity to low micromolar activity against the purified protein. So this is an orthogonal validation that the yeast is telling us the truth. And so diving quickly into mechanism, when we looked at this compound, it looked to be a potential covalent inhibitor. And so we thought that this compound was actually forming an adduct with the catalytic cysteine. So to quickly test that, we did mass spec on the purified protease itself and in the presence of compound. And what we saw was a nice approximately 95 Dalton shift, which is exactly what we expected if the compound was binding covalently to the catalytic cysteine of the protease. And so we've been working with Larry Shapiro at Columbia to hopefully crystallize the 3CL protease bound to this compound. And this is some of the initial work that he's produced where suggesting maybe hints that we can possibly crystallize this interaction. So next steps for this project, we're screening additional compounds. We're, again, part of this large AViDD consortium. We're trying to further develop some of our other inhibitors. We're not actually focusing a ton on the 3CL protease at this present time. There's lots of other hits we've been pursuing and further developing. And we were interested in applying our platform to other essential viral proteins outside of proteases. So sorry for part 2 of our talk. Described how you can use DNA barcoding to study lots of models at once. Despite the scale we're operating at, you get real hits. And then we validated one of our sets of hits as being broad-acting as we were looking to discover. So with that, I'd like to always thank a few people. Obviously, I'm nothing without the amazing people I work with. And there have been some really awesome graduate students and collaborators for the protease work that we've done, and particularly Sho, Debbie, Bruce, and Sam, along with David Ho, who has been just an amazing collaborator. And also, I'd always like to thank all the other members of my lab that make coming into work really, really great, and the funding that helps enable all this great work.